Hello, everyone, and welcome to Ask the Expert. I'm Alexis Carmer. I'm standing in for Monica today. It's my pleasure to welcome Marika Falcone today. She's an MD, PhD coming to us from Milan. Her research career has been focused on understanding the pathogenic mechanisms underlying autoimmune diseases. After completing her MD degree at the University of Rome, she completed her PhD thesis work at Einstein and her postdoc at the Scripps. She then returned to Italy, where she's been working as a PI at OSR for the past 2 decades. Since April 2021, she's been directing the autoimmune pathogenesis unit at OSR. In the past decade, her research interest has been focused on studying how the intestinal environment regulates the pathogenesis of extraintestinal autoimmune diseases such as T1D and MS. Her research group has made important contributions on understanding how the crosstalk between the gut microbiota and the immune system triggers autoimmunity in extraintestinal organs such as the pancreatic islets in T1D and the central nervous system in MS. Thanks to a close collaboration with OSR clinical departments, she was able to collect human intestinal tissue samples from individuals affected by T1D and MS and link alterations of gut mucosal immunity and microbiota composition with the occurrence of autoimmune diabetes and brain autoimmunity in humans. In addition, her team has determined the fundamental role of the gut barrier in regulating the crosstalk between immune cells and the gut microbiota and preventing inflammation in the extraintestinal tissues. Marika, thank you so much for sharing your work today. I'll turn it over to you now.
Thank you. Thank you for the nice introduction. And I would like to thank the Sugar Science for this very nice, kind invitation. So today I will present our work that try to address if and how autoimmunity against the pancreatic islets originate in the gut. Okay, so this is the topic that I will address today. And I will start talking very briefly about what we know about the pathogenesis of autoimmune type 1 diabetes. And this to let you know how and from where it came to our mind to the question whether the pathogenesis of the disease originates in the gut. And then I will show you some evidence, in particular that we collected in humans affected by type 1 diabetes, showing that the gut may be an important hub where the autoimmunity against pancreatic islets take place. And then I will go into our work that was published a couple of years ago in PNS, showing in an experimental model of type 1 diabetes, the transgenic PDC 2.5 mice, how intestinal activation of beta cell reactive T cells can take place in the gut through an abnormal crosstalk with the gut microbiome, and how this ultimately will lead to type 1 diabetes. And then very shortly at the end, I will talk with you and I will discuss to you what are the implications of all these findings for prevention and treatment of type 1 diabetes. So let's start with what we know about the pathogenesis of the disease. So we know that prevention of autoimmunity mostly takes place in the thymus, so very early on in life. So what happens is that T cells that are specific for self-antigens, so antigens and that belongs to our body, they are deleted in the thymus, and this to prevent the immune system from attacking our own components. For example, the pancreatic islets in the case of type 1 diabetes. And so we know that in diabetes, there is a failure of this thymic mechanism. And the consequence is that some islet-reactive T cells enter the peripheral blood circulation and the lymphatic circulation. But what we still don't know how these T cells are activated in the periphery, and so why at some point they decided to enter the pancreatic islets and destroy the insulin-producing beta cells. I was mentioning the importance of the thymus. So why the thymus is so important? We know that in the thymus, many antigens, so protein of our body that are expressed in the peripheral organs, they're also expressed in the thymus. And these induce a deletion of T cells that are specific for these components. For example, insulin is expressed in the thymus, and the consequence is a deletion of most of T cells that are specific for insulin. However, and at the same time, there are some regulatory T cells, so T cells that are capable to suppress autoimmunity. These cells are generated in the thymus and are specific for insulin. So in case there is a response, for example, against insulin, these cells, their function is to suppress this response. So these are very important mechanisms, and we know that in diabetes, in human diabetes, these mechanisms can be failing. For example, we know that if we look at the genetic risk to develop type 1 diabetes, one of the major factors related to the genetic risk is the presence of an alteration in the insulin gene. And so this genetic alteration can ultimately lead to a defective expression of insulin in the thymus. So these mechanisms of prevention of autoimmunity can be failing in individuals that are genetically predisposed to autoimmune diabetes. However, the fact that some individuals may have reduced number of regulatory T cells specific for insulin or other islet-specific antigens is not or the fact that there are some naive T cells that from the thymus go into the peripheral circulation is not sufficient to induce diabetes. This because in order for a naive T cell specific for islet autoantigen, in order to go from the peripheral circulation into the islet and destroy the beta cells, they need to be activated at some point. Activated means that their T cell receptor is triggered, so they see their specific islet autoantigen presented by dendritic cells, and at the same time, they acquire an effector phenotype, so the capacity to secrete inflammatory cytokine, but also to acquire some killer function through which they can directly destroy the pancreatic islets. So what is still an open question, and let's say a mystery in the field of autoimmune diseases in general and type 1 diabetes, is what activate the islet-reactive T cells in the periphery. And we believe, and this is why we are looking into the gut, that the intestine can be an important site where the islet-reactive T cells get activated. And this hypothesis really originated from several findings that have been collected in the last, I will say, 10, 15 years, showing that the gut is a very important site for modulation of immune cells. So not only autoimmune T cells involved in type 1 diabetes, but all immune cells at some point in their life go through the intestinal mucosa where they can be modulated. And this is important for all the diseases that are regulated by the immune system. It is important for the response against infectious agents, it is important for anti-tumoral immunity. And so this is a field that is expanding enormously in the recent years. And we believe that at some point also islet-reactive T cells circulating through the gut mucosa, they receive a modulation from the gut environment. So from the microbiota, but also from some dietary components or any other environmental factor that change the intestinal environment and has an impact on the immune cells that at some point are present in the gut mucosa. So what we think happened in autoimmune diseases is that in normal conditions, so when the intestine is not inflamed and contains a normal, let's say, beneficial gut microbiome, and the gut barrier, so the layer of intestinal epithelial cells that separate the microbiome from the immune cells is, let's say, normal, what happens is that the immune cells that circulate through the gut mucosa, here they receive some tolerogenic signals, so they become regulatory T cells. And then from the intestine, they can travel to peripheral organ where they can actually suppress autoimmunity. This has been demonstrated not for type 1 diabetes, but for other extraintestinal autoimmune diseases, such as the experimental model of multiple sclerosis. However, what we know and what we learn in the recent years, that in case the gut is inflamed, and for example, because there is a pro-inflammatory gut microbiome, but also for other reasons, for example, some dietary components, and we will talk later, can directly induce a damage of the gut barrier and inflammation in the gut. So all these environmental conditions may trigger a damage of the gut barrier, and as a consequence, the gut microbiome can come in contact with immune cells, including self-reactive T cells circulating through the gut mucosa. And what happens is that these induce an activation and acquisition of an effector phenotype by these T cells, including islet reactive T cells. And from the gut, then they can move to peripheral organs where they can induce autoimmune diseases, including type 1 diabetes going to the pancreatic islets. This is all very nice. What evidence we have that this is really what's happening and in humans and in experimental models? First of all, of course, I will present what we know in humans that I believe is relevant for the disease. And what we know, and we have been knowing since 2006, is that individuals with type 1 diabetes, they have an alteration of the gut barrier integrity. So they have an increased gut permeability. And you can see in this study that was published by our clinical department here at the San Raffaele Hospital, it was shown that patients with type 1 diabetes compared to control have an increased excretion of lactulose mannitol in the lactulose mannitol test, meaning that they have a damage of the gut barrier. And this both new onset and long-term patients. However, what was very interesting that also individuals that still have normoglycemia, so they haven't developed yet type 1 diabetes, but they have autoantibodies against GAT65, insulin, or other allet antigen. So they have autoimmunity, but not yet diabetes. They also have an increased gut permeability. So this suggesting that this gut inflammation and gut phenomenon is somehow associated with the pathogenesis of the disease. More recently, we collected more evidence showing that there is a damage of the gut barrier integrity in type 1 diabetic patients, so an increase in zonulin, but also an increase in some markers like the LPS binding protein and the MD2 that show that there is bacterial translocation, so that some components of the gut microbiome travel from the intestinal lumen into the gut mucosa and circulation. And then in humans with type 1 diabetes, we also have evidence that there is alteration of the gut microbiome. Here is the list of the studies that were published up to 2018 but there are many more. But just to show you that now the fact that patients and also individuals with autoimmunity, so not yet diabetes, compared to healthy control, they have alteration of the gut microbiome. Not always the same, but there are some that are recurrent in the different studies. For example, there is a degree of protective anti-inflammatory species like the Prevotella and the Clostridia, and a decrease of short-chain fatty acid producing species, while there is an increase, for example, of Akkermansia that degrade the mucus layer. So it's important because affect the gut barrier integrity. And recently, we also performed a study looking at gut mucosal immunity in patients. So we had the opportunity, and we started more than 10 years ago, to collect intestinal biopsies from patients with type 1 diabetes. And in these biopsies, we perform a FACS analysis to look at the immune cells and their functional phenotype. And at the same time, we perform the microbiota analysis in this tissue. And what we found early on, we found that there is a decrease in the presence of regulatory T cells in the patients. And more recently, we also found there is an increase in the effector phenotype. So they produce a lot of IL-17 and IL-22 and TNF-alpha. And this increase in the pro-inflammatory phenotype of immune cells is associated with alteration of the gut microbiota composition. So as I say, all of this is very nice in humans. So let's say, suggest that effectively the gut environment may be important for the pathogenesis of human type 1 diabetes. However, all these findings are correlative. So we don't know whether these alterations somehow are involved in the activation of beta cell autoimmunity and onset of type 1 diabetes. So in order to look and find a causal link between these intestinal alterations, so dysbiosis, gut barrier damage, and alteration of gut immunity, and the pathogenesis of type 1 diabetes, we went and studied preclinical models, animal models. So as I told you at the beginning, we know that T cells specific for islet antigen escape the thymus. We still don't know what expand and activate them in the periphery. So in order to answer this question, we decided to use the T cell receptor transgenic BDC 2.5 mice. And I will briefly explain what are these mice. These are mice that were generated now 30 years ago, but they are very interesting because as individuals that have islet-specific T cells escape from the thymus, they have a large population of T cells specific for islet antigen in the periphery. How were they made? What they did was to take total immune cells from the spleen of the NOD mice, that is the spontaneous model of type 1 diabetes, and at the same time, they took islet extract containing all the antigen of the islet, and they put them together in some well, of course, in culture condition, and they derive some clone. What are the T cell clone? These are, let's say, from each well, they put less than 1 cells. So at the end, the cells expanded responding to the islet antigen, and from each well, they recover 1 clone, so meaning all the T cell in that well derived from 1 single cell. So from all these clones, they derive this clone called BDC 2.5. They amplify the T cell receptor, and they generated some transgenic mice that are transgenic for this specific T cell receptor, recognizing an islet antigen. So in these mice, as I told you, 90% of T cells are specific for an islet antigen, and we believe this somehow is similar to the condition of individuals that have islet reactive T cells in the periphery. Importantly, in these mice, you don't have diabetes because, as I told you, these cells are naive, are not activated. So what they do, they go into the pancreas. You can see here the picture. The dark cells are the immune cells, and the white body is the pancreatic islets. You can see this, the immune cells surround the pancreatic islet, but they don't destroy them because they are specific for islet antigen, but the cells are not activated. So what we did in these mice, actually with what Chiara Sorini did, that was the PhD student first and then postdoctoral fellow that performed those experiments, what she did was to induce chronic colitis in these mice. So in these mice, we induce a damage of the gut barrier to test whether this can affect the activation state and functional phenotype of the islet reactive T cells. And so we induce this colitis, and you can see here, normally what you do is to weight the mice to check that they lose some weight. You check their colon, the normal, usually shorten with the colitis. And importantly, in these mice, we had the damage of the gut barrier that we measure through expression of the tight junction protein, and we have an increased gut permeability. And so, interestingly, in these mice, what happened is that the islet reactive T cells that we can trace because they have a V-beta-4 chain, so that we can detect through a specific antibody against these T cell receptor. So what we detected is that in these mice with colitis, with DSS, that is the agent we use to induce colitis, we had an activation that we measured through expression of this CD44 marker. And we have this activation in the clone, but also in all the T cells in the gut mucosa, of course. And not only they were activated, but they also acquired an effect of phenotype. So they were secreting IL-17 and interferon gamma. Importantly, not only in the gut mucosa, but also these islet reactive T cells isolated from the pancreatic lymph node and islets, they express marker of activation, and they also secreted large amounts of IL-17. As you can see here, we use this alpha-4 beta-7 marker that tell us whether these cells are coming from the gut or not, because they acquire this marker when they go to the gut. And as you can see, cells that we found in the pancreatic lymph node and within the islet, so the intra-islet lymphocytes, they express this marker that told us that were coming from the gut mucosa. What was most important is that these cells now, these islet reactive T cells that are activated in the gut and travel to the pancreas, they are now able to destroy the pancreatic islets. So you can see here the control, again, the immune cells surrounding the islets, while in the mice with colitis, the immune cells enter the islets. You can see all dark cells within the space where the islet was before, and so the islets were completely destroyed. And as a consequence, these mice, 60% of them develop autoimmune diabetes. Another experiment we did in these mice is to test whether the microbiome was important for this activation of the islet reactive T cells. And what we did was to treat the mice with colitis with antibody to remove the gut microbiome. And as you can see in these mice with colitis, DSS colitis, when we added the antibiotics, these mice were completely protected from the disease. So meaning that somehow the gut microbiome was involved in this activation of the islet reactive T cells. However, the gut microbiome was not sufficient, because if we took the fecal material from the mice with colitis and transfer into naive mice, these mice did not develop diabetes, meaning that it was not the gut microbiome itself, and without the gut barrier damage induced by the DSS and the colitis, we did not detect any autoimmune diabetes. So of course, the next question was how the gut microbiome triggered the activation of the islet reactive T cells. And to answer this question, we performed a very simple basic experiment. So we took the whole murine microbiota from an healthy or mice with colitis, and we added to the BDC 2.5 islet reactive T cells in vitro. And you can see here, these are the cells unstimulated. In red, the cells in which we did not add any microbiota. And in green, the BDC 2.5 T cells with the gut microbiota from a control mouse, so a mouse without insulitis. And even the normal murine microbiota can induce activation of the BDC 2.5 T cell clone. The microbiota from the colitis mice was better, but not much difference we detected. And importantly, we demonstrated this mechanism is T cell receptor mediated. So when we block the presentation of the islet antigen by the dendritic cells in culture, giving an anti-MHC class II antibody, we did not detect any activation of the clone, meaning that the microbiome was able to activate the islet reactive T cell with a T cell receptor mediated mechanism. And as you can see here, there are other studies showing that the gut microbiome can trigger the activation of islet reactive T cells, different clones, not just a BDC 2.5, through mechanism of molecular mimicry. And so now we have to demonstrate that this is also taking place in humans. What we have been doing recently is to search whether these molecular mimicry mechanism can also take place in humans. What we did was an in silico screening, testing whether there is homology in sequence between the self-antigen, the human self-antigen, the most known in human type 1 diabetes, such as insulin, GAD65, GAD62, and chromogranin A, and the sequence of all the bacteria that are present in the healthy human commensal microbiome. And the results are quite striking because, for example, you can see that from chromogranin A, that is also an antigen that is expressed also by the intestinal tissue, there is a very high alignment between several bacteria that are present in the normal human microbiota, but also for other antigen. For example, the non-specific islet cell antigen, there is a homology 100% with many bacteria, and also the GAD65 peptide 352. So this is just a preliminary analysis, but to tell you that this phenomenon of molecular mimicry can be something that takes place quite commonly. And so the gut barrier, the leakage of the gut barrier, so the abnormal interaction between the gut microbiome and immune cells, including islet reactive T cells in the gut, can be responsible for activation and final triggering of type 1 diabetes. If I have still a couple of minutes, I want to just briefly talk about some important implication of those findings for type 1 diabetes. One implication is that, of course, if we are able to restore let's say, a normal, non-inflammatory gut environment. So if we are able to, let's say, treat and prevent any leakage of the gut barrier in humans, particularly in individuals that may have high genetic risk to develop type 1 diabetes, through diet, for example, or other intervention, we will be able eventually either to delay onset or to prevent at all the onset of type 1 diabetes. And how can we restore a healthy gut environment? As I mentioned, mostly through diet. We know that there are dietary components that are very important, either to induce a pro-inflammatory environment or to maintain a tolerogenic protective environment. For example, emulsifier, they induce gut barrier damage and gut inflammation. And while there are other components like the indole-derived and tryptophan-derived ligands that are contained in cruciferous vegetables that are protective for gut barrier integrity and reduce inflammation. Also, omega-3 and dietary fibers are protective, for example. And so what we did to try to see whether we can restore gut barrier integrity and prevent diabetes through diet, we did some study in the NOD mice where we administer a diet which enriched in soluble fibers, in particular inulin and omega-3. And in these mice, we found the protection from type 1 diabetes that was associated with, let's say, a protective gut microbiome environment, so an anti-inflammatory metabolic environment, and a reduced presence of inflammatory cells and increase of regulatory T cell differentiation. And of course, now we will try to translate all these findings into something that can be useful for humans. And what we are doing now is we started, thanks to a European grant, to test whether a specific diet that targets the microbiota, so try to restore an anti-inflammatory gut environment, can be beneficial in children with type 1 diabetes. So we are actually now building a platform in which all information about the gut environment will be integrated with the genetic profiling of the children and also information regarding their diet in order to, on one hand, measure the risk to develop type 1 diabetes based also on the microbiota profile, and then eventually do some intervention in their diet in order to improve their beneficial microbiome and reduce intestinal inflammation and gut barrier damage. And I finish. I just would like to thank all the people that were involved in this study. In particular, I would like to thank the clinical unit of the San Raffaele Hospital, because they were instrumental to provide us with human samples. And also, I would like to thank the Center for Omics Sciences and Metabolomics Analysis and our external collaborator that were really helpful to carry out this study. And thank you for all your attention, and I'll be happy to take any questions.
Thank you so much, Marika. That was really interesting. You did an excellent job of putting together the puzzle pieces to help us understand, how you've investigated this question of the microbiome and type 1. That was really interesting. Thank you. You can, if anyone has a question, please unmute yourself or go ahead and throw it in the chat box, and I can read that. While we're waiting for that, Marika, I did have just a couple general questions. One is, I noticed in your papers, some of those clinical samples, I think, were not only from type 1 diabetes, but your group also studies MS and some other autoimmune disorders. I'm just curious if you could talk about some of the similarities and differences between the MS and the autoimmunity and the gut factors compared to MS and type 1 diabetes.
Yes, thank you for this question. We believe that the gut can be, let's say, a common denominator in these diseases, because of course we know that they are multifactorial, so there are many, many genes that are involved. And of course, this genetic predisposition may be very different between, of course, type 1 diabetic patient and MS patient. However, at some point, when you have these autoreactive T cells circulating that, of course, in MS are specific for myelin antigen, in type 1 diabetes are specific for islets, when you have them and they go through the gut, there in the gut, they may have a wrong education, a wrong activation, acquisition of an effector phenotype. we found some difference in humans with MS and type 1 diabetes in the gut. Particularly in MS patients, we found mostly an increase in Th17 cells in the gut. And we know that in MS, Th17 cells are crucial for inducing damage of the myelin in the brain. While in type 1 diabetes, more than an increase in Th17 that sometimes we also found, we found an increase in Th1, and mostly we found a decrease in regulatory T cells. This is something that we detected very early on. And so we believe that also what's happening in the gut may be different between type 1 diabetes and MS, because also there may be some genetic factor that also alter the capacity of individuals to react to the insult in the gut. So there may be also, this is something that we are also investigating, some genetic predisposition so that the intestinal barrier may be more susceptible to the inflammatory insult coming from the microbiota, the diet, or any other environmental factor.
Go ahead, Imra.
Yeah, thank you. Dr. Falcone, nice to meet you. I also had my PhD in Italy, so it is great to see an Italian scientist. So my first question is related to your data coming from the intestinal samples. So you see a very big difference between the controls and type 1 diabetes patients. Were they age and sex matched? Or even though they were not matched, the difference was so clear? And my second question is, which part of the intestine did you collect these samples from? Small intestines, duodenum, jejunum, ileum, or colon?
Thank you for the question. So to answer the first question, we try to match for sex and age. So these are all, let's say we have two cohorts. Some children that they had the suspect of celiac disease, and then they turn out to be not celiac. And we have some adults, so we try to match as much as possible. And of course, they are the mean age is similar between the patients and the control. These are the one I show you today are from the small intestine, so the duodenum. And the reason is that in animal models, the small intestine is really the site, for example, where regulatory T cells are differentiated, and also where most of the immune cells interact with the microbiome. But of course, also the colon is very relevant because you have much larger microbiome species and diversity. And so it's also relevant to study the colon. And for example, the Clostridia induced direct cell differentiation in the colon in mice. So the colon is also a site that we are interested in study, and we hope to be able to do that. As you can imagine, the machinery to collect intestinal samples from patients that don't have any intestinal disease is not so easy. No. But in the next year, we will do that.
Mm-hmm.
It looks like we have a question in the chat box. Did you determine if any specific gut microbe species in BDC 2.5 mice treated with DSS mice versus controls could be responsible for T1D if tested in mice?
We didn't do that, and the reason is that, yeah, that's another important question. It's not easy because that's the reason why we did the in silico analysis, to see whether there were only few species that show homology of sequence with the islet antigen. When we saw that there are so many, we don't think it's just one single bacterial species, but there can be many different species. So it's true that we know that the antigen that triggered the BDC 2.5 clone is a peptide of chromogranin A. But as I show you, there are several bacterial species that have homology of sequence with that chromogranin antigen. Yes, so we didn't do that for this reason.
Marika, I'm curious. You an earlier question was about where the samples were taken, and that was a question in my mind too, considering most of the information we have about the human microbiome, especially in type 1 diabetes patients, is, I assume, from stool samples, so obviously from the colon. I'm just wondering, moving forward, trying to gather more information about autoimmunity and the gut microbiome. Has there been a standardization of collection methods and ideas about the data you're collecting in humans that will help kind of collaborate and put all this information together?
Yes, we have been doing this collection. We started collecting the microbiota from the intestinal tissue, and this was somehow misleading because then you have a lot of host DNA and can be, let's say, wrong. And then thanks to a collaboration with Bana Yabri that has a large experience in children with IBD and celiac disease, or inflammatory disease of the gut, she had been developing a technique to collect the microbiota during esophagogastroduodenoscopy through brushing of the gut mucosa. So in the last years, so the data I present you today, and they are in the paper that we recently published in Biomedicine, they are actually taken with this brushing method that is also used in inflammatory bowel disease, in celiac disease. In type 1 diabetes, I'm not aware of any other group that does this type of study in humans. But of course, the protocol is standardized. It's used in other clinical settings.
Thank you. Emrah, did you have another question?
Yeah, I have a follow-up question. Because these samples are so precious and important, we have been using fecal samples, which questionable if they really represent what is going on in the gut. So did you use these samples on NOD mice or germ-free NOD mice to determine if they can trigger the type 1 diabetes onset? And do you still have these samples?
Yes, we do have them, and part of them we should have some left over. Regarding the experiment of the, let's say, humanized microbiota mice, we did. What we actually performed was a fecal transfer from individuals with type 1 diabetes and healthy control, but we use fecal material. The reason is that we didn't want to waste the one from the small intestine. But this experiment was quite difficult because we actually transferred in the BDC 2.5 model. So our goal was to induce gut barrier inflammation and damage and see whether this is sufficient as the DSS to induce diabetes in these mice. However, it's very difficult. I mean, in our hands, it was very difficult to obtain colonization of mice with the human microbiota. It's true we did with antibiotics. We don't have a germ-free facility here in Italy, actually, not just in Milan. And we will try to do that thanks to a collaboration with the hospital in Berlin. But we haven't done that yet. But it's our intention to do in BDC 2.5 as a recipient, and germ-free possibly, and using also the microbiota from the small intestine.
Okay, I think that's the end of our questions. And in the interest of time, we'll close. I want to thank you again, Marika, for your presentation, your wonderful presentation. Shortly, it will be available on the Sugar Science YouTube channel and our podcast for others to enjoy as well. And we look forward to hearing more from your group. Thank you so much.
Thank you. Please contact me if you want any question. I have my email at the beginning of the presentation.
Thank you so much. Bye-bye.
Bye.